Good afternoon, everyone. You are warmly welcome to the Department of Biochemistry, Cell and Molecular Biology, and especially to the WACCBIP conference facility, which you are sitting in now. We are here for a very special occasion. We are here to remember one of our very own, pace setter in the world of sciences, particularly for women in science. We thank you for availing yourselves and making time to come here today. So to start off the program, we are going to have a short prayer by one of our students. I would like to call on Mr. Ewurama Addy to come and open this.
Our Heavenly Father, we thank you for today. We thank you for a day like this in the Department of Biochemistry. We thank you for the Marian Addy Memorial Lecture. We commit it into your hands and we ask that you shall run it to a successful end. In Jesus' name we pray thanksgiving. Amen.
Okay, so now that we've prayed, let me properly introduce myself. My name is Dr. Lydia Mosi. I am the current head of department for this Biochemistry, Cell and Molecular Biology department. By way of announcing the purpose of this gathering, I would like to just first and foremost invite key persons who are supposed to be on the high table to please join me up here. So without much ado, Mr. Provost, can you please join us up here? Acting Dean, Physical and Mathematical Sciences, please can you join us up here? Reverend, please can you join us up here? And Mr. Okuadi, if it's possible, can you please join us up here, unless you prefer to sit down. So, it's been 5 years since the passing of our dear Professor Marian Ewurama Addy, and this is the second edition of the lecture series that we hold in her honor. The first one was held on March 4th, 2015. By way of history, this department was started in 1963, formerly as a part of Nutrition and Food Science Department, and later as a standalone department in 1970. Now, Prof Addy joined the department in 1977, and during that time, she served 2 consecutive terms as head of department from 88 to 91 and 93 to 98. More importantly, she became the first female full professor in the sciences whilst she was here at the department, and was also elected as a fellow of the Ghana Academy of Arts and Sciences. She retired in 2002, but continued to teach on contract and part-time basis until 2008. I joined the department in 2010, and by way of adding myself to history, I inherited her desk in the old department, and I moved it to this department as well. So, yeah. I've made my own small full stop there. So all the while, she still continued to support the department throughout the time until her passing. Now, everyone knows Prof as a very passionate scientist with a very deep interest in medicinal plants. She also did extraordinary work to demonstrate how these plants and herbal preparations could be used in drug treatment, in disease treatment, and to also figure out their potential toxicities whilst they were being used. She absolutely loved teaching. Unfortunately, I wasn't taught by her, but she does have quite a few people in this room who she taught. She used to refer to everyone who came into her class as her children, I remember. Her love for sciences was really, really evident in her activities. Everyone knows her, even if you were a baby at that time and now you're growing, everyone knew her as the quiz mistress for the National Science and Maths Quiz. Back then, I think it was called the Brilliant Science and Maths Quiz. I don't even know whether Brilliant still exists. It's a soap that we used to use. Now it's been ably run by Dr. Elsie Kaufman of Bioengineering. She was very, very passionate about the role of women in science, and it's actually very important to note that because last week Friday was a day where for International Women in Science and International Day for Women, and we celebrate all her roles that she has played. So today, as we mark an anniversary for her, we, as the department, in conjunction with her family, have put together this celebration to exhibit and celebrate all her extraordinary achievements, particularly her outstanding contributions to scientific knowledge. Today, we would want to do things the Prof Addy way. Unfortunately, we didn't start at 4 o'clock as she would have started exactly at 4 o'clock. But for the science talk, we'd like to make it very simple. And I think Reverend would do a very good job of that. We will have a teaching session. And in that teaching session, Reverend is going to give us evidence-based medicinal properties of kombucha drink. I am honored to be part of this program, promoting the multidisciplinary role of a woman in science. And I would like to most importantly warmly welcome her family who have all joined us here today, and also the family of Reverend W.S.K. Gbewonyo, who are pretty much here for what I would call his inaugural. We would also like to ask if it's not too much of a problem to have her immediate family stand so that we can recognize them at this moment. Her immediate family, please, if you're here, can you stand and let's welcome you. Thank you very much for coming. Thank you. Thank you very much. So without much ado, I would like to continue this program by calling on one of our own, Dr. Anastasia Akins, to introduce the chairperson for today's event. Thank you so much.
Good afternoon, our distinguished guests. I deem it an honor to be asked to introduce the chairperson for today's occasion. He is a professor of geology and an internationally recognized researcher in the field of sedimentary geology. He has 18 years university teaching and research experience. He has undertaken several extension activities, mostly in the areas of education, research, and consultancy services. He holds a bachelor degree in geology and a PhD in geology, and he also holds a postdoc certificate in offshore prospecting. He is currently the provost of the College of Basic and Applied Sciences. Ladies and gentlemen, I'm sure at this point most of you know who our chairperson is. He is in the person of Professor Daniel Esiedu. Let's welcome him. You are welcome, Prof.
Thank you, Dr. Akins. You would notice that on the programs you have, there was supposed to be a short documentary on Prof Ewurama Addy. Now, this documentary had been running all throughout, and for the sake of time, we will just move on. And then, after the event, if people still want to watch it, we would project it for them. At this juncture, I would like to invite the daughter of our speaker, Yesuto Gbewonyo Amedopo. She's going to give us a poetry recital.
Once again, Asasiya awakens from her slumber. Seeds planted into soils of time spring forth. Roots wander, tree backs shudder, and leaves rustle at the squall of the new dawn. An heirloom of knowledge as vast as the Etiwa forest of yesteryears become the canopies under which we sit. Its trees providing the luscious fruits we eat, not only for today, but for years to come. For once upon the many years before this day, an herbalist in white coat came to play, with a pipette of modern medicine in one hand and beakers of indigenous Ghanaian knowledge systems in the other. She calculatively brewed her concoctions, filtering them with various flasks for appropriate consumption and transferring them through the appropriate funnels of communication so that our faith was renewed in traditional ways of healing. Oh, Ghana and Africa really do have a lot to offer the world. No longer would plant medicine be confined to the cradles of the witch doctor or drunk only from the calabash of the villager. No longer will it serve as alternative medicine only for the poor. No, for it has gone through the doors and corridors of the lab, tried through the Bunsen burner, proving its worth as with gold. Is it asthma? Drop your inhaler. For from the smoke drum chymographs to physiographs and ELISA, this herbalist proved the efficacy of extracts from our very own Anansinkatia, Desmodium adscendens. For type 2 diabetes, the herbalist proved how extract from Indigofera arrecta would excite the islet cells to release more insulin when blood glucose levels began to rise high. And in the case of malaria, we give credit to her works on Cryptolepis sanguinolenta, for which reason we can safely find healing in drinking Nibima. Need I say more? The list goes on. And day by day, we witness these herbs come on display in the modern pharmacies in carefully calibrated quantities, a gesture that embraces their validity. Yes, this herbalist said rightly, there is health in us. Today, we remember her, Professor Marian Ewurama Addy, the herbalist who wore a white coat and yet was not colonized by it. The one who put science into the art of healing with herbs and emancipated traditional plant-based medicine from the shackles of dark age reasoning. Heeding Nananum when they said that knowledge like a baobab tree cannot be embraced by one person alone, she studied at the feet of our traditional practitioners and projected their wisdom to the world, sculpting it in the very art of scientific reason and fact. Partnering other scholars of science, she upheld the understanding that the strength of the broom lies not in the power of a single frond, but in the resilience of its united fronds. So today we are here not because she worked in isolation, but because she freely shared her insights with us all, drawing us and the rest of the world into acquaintance with the healing gifts of nature dotted in our backyards and scattered abroad in our fields. We remember her exudation of Ghanaian identity as she stood before the world, clad in traditional Ghanaian raiment. Her unique head wrap and kente stole. A true daughter of the land and woman of noble character. Professor Marian Ewurama Addy. Your footprints have set the pace in the space for many, especially women, in biology, chemistry, botany, biochemistry, agriculture, and other natural sciences. Many who would go on to appreciate the seeds planted in the soils of time. Many who would preserve this heirloom of knowledge in our daily pursuit for health and well-being. Professor Marian Ewurama Addy. The herbalist in white coat, who stood tall with a pipette of modern science in one hand and beakers of indigenous Ghanaian knowledge systems in the other, calculatively brewing concoctions, filtering them into various flasks for appropriate consumption, and transferring them through the appropriate funnels of communication so that our faith was renewed in traditional ways of healing. See how your legacy lives on. Thank you.
Biochemistry and poetry. Hey. I think we all need to give her another round of applause. So without much ado, I'd like to call on the chairperson to introduce the speaker. Prof.
Good evening, ladies and gentlemen. I'm sure all of you have this brochure, and it has the biography of the speaker, Reverend Dr. W.S.K. Gbewonyo. So I will not bore you by reading this, but I'm told that he is a student of Professor Ewurama Addy. And he is going to give us a lecture on the title towards sustaining Marian Ewurama Addy's legacy, evidence-based research on kombucha as a health drink. Reverend Dr. W.S.K. Gbewonyo is a senior lecturer at the Department of Biochemistry, Cell and Molecular Biology. Prof, please come in and deliver your lecture. The audience are waiting for you. Thank you very much.
So good evening and welcome again to all of us. Now I haven't seen for I don't know how many years, decades. Those days that Prof Addy had them at the basic school, and they were coming to the department, and together with Auntie Year's children. So we had a whole company, and we've known each other for a while. Prof Addy, more or less, I've been her student, but since I got married, more like a family, because the mother was a baker, she loved only one bread from one source, and that is from my wife. So it's a delight to have this opportunity to present this lecture in her honor. And I thank the family for coming over once again and giving us the opportunity as a department to offer this. Sustaining Marian Addy's legacy, evidence-based research on kombucha as a health drink. I know that many of us here haven't heard this before. They don't know what kombucha is, but the very older people who may probably have heard about kombucha. So I decided to start with this because that's about the only picture we have that shows Prof Addy here, together with Prof Odro in the middle. At that time, he was the head, Prof Jan, and then two of us who were the youngest to join them, Antia and I. In fact, we joined the department a week apart in October 1980. So, and at that time, we had only 12 students. Only 12 students. And from that period on in the 1980s, it's only these 5 of us until Prof Okain, Dr. Ajimani, and Prof Okain and Lord and Prof Saki, them joined us. And since then, we managed to have a number of students come out, and in the 2000s returned. And towards the end, I will get back to that also, because they have made very extremely significant contribution, including providing a place like this for us to have a lecture. So we're talking about sustaining Marian Addy's legacy. And what indeed is that legacy we are talking about? What she did was to authenticate claims. The poem has said it all. I don't know, she is a sociologist. I don't know how she put all this together. I'm surprised how she got all, of course she did some science, but basic science in the secondary school. But thank God for that presentation. So that poem said it all. She authenticated or tried to authenticate the claims to efficacy. The herbalist will always say, you know, this herb is efficacious. And then we see them. They bring them, people are buying, one of my colleagues, Prof Marian, couldn't make it. He is a toxicologist. And they bring it to him, and he said, this one is a killer, and yet it's on sale. And you wonder. And some of us here, of course, patronize this because of these claims that are made. So Prof. Addy's aim was to authenticate the efficacy of these herbal remedies. And not only that, but to encourage these traditional practitioners to subject these remedies to verification of their claims. For verification of their claims. And not only that, but to also serve as a bridge between the clinicians and the practitioners. Because it's like these two normally don't meet. But at least one person tried to make an effort, and that is Dr. Kwampofo. And I had the privilege, of course, of through Professor Addy to interact with him as a very young person. So what she did was to carry out bioassays. Bioassay means we take the whatever product it is and look for a medium that will more or less imitate whatever action is taking place within. And then we expose that medium to it and get some response that we see, which is very similar to what they expect to happen. So that basically is what we do. And we can carry this out in animals, most often because we are not allowed generally to do this in humans until some approval is given from some wherever quarter. So she did her best to bridge that gap. Now, in 2015, Professor Nyaku, who I think he was the third, Dr. City is here, so the two of them, I was the first, and they came together after Dr. Aume, or Professor Aume, who is now in Canada. So he gave the first lecture on evidence-based research into plant medicine, the Marian Ewurama Addy factor. And during that lecture, he enumerated and educated us on a lot of the work that she had done. Now, Marian Addy's mentorship, because she mentored me, and she, of course, will supervise, and that supervision included ensuring that you did everything very well. Sometimes, I think I went to an extent, and she said, you want to be a perfectionist. And I didn't want to move, and it takes time. She had to push for me to get things done. And by the way, I mentioned Dr. Kwampofo, so she will take me in there was a two-door car. I don't know whether it was an Opel. Yeah. And so the two of us will be in it and make trips to Mampong. Go and see Dr. Kwampofo. And I think it was one or two, I think at least one occasion, we went also to Dr. Namasi to visit him. And, well, thank God for her background as a botanist to start with. She did botany and chemistry. So the plants, instead of somebody else collecting it, and I collect it fresh. So she took me to Bury Gardens and showed me how to get these plants myself. And Professor Ite Smith, those who read or who had read her book, Rewards, will see one of the things she mentioned is how she will go around departments picking bits and pieces of equipment. And one of those was an equipment we got from Professor Ite Smith in Kolebu. After we struggled and were trying to, after I went and got some model we could use, we had to go to him because we had to be smoking drum. That means you had either a candle or, this is our, wick that had, you have kerosene and then it will be smoking. So we use this and kind of blackened paper, put on a drum, the soot, and then use that to trace. We were doing this, and then she found that she could get some physiograph from Professor Ite Smith. And so eventually, my work, I ended up using, instead of the chemograph, used a physiograph. So my thesis, at some point, I thought I still had a chemograph in it until I went back and looked at it. She encouraged me, and I went to Prof Opako at the University of Ibadan. And I had to go there because the model we were using, he was the one who brought that up. So I went there and learned from him. Now, she encouraged me to go to presentations, and there was this one at Ile-Ife, and she arranged for me to go with Dr. Namesi, Professor Ayim. So there was a group from Tech together with Dr. Namesi, and we drove in Dr. Namesi's Benz, those days, that Benz. And so we went through from here, Togo. Those who have ever been on that route, early 80, you can imagine what we went through, the gendarmes and the bribes. So it took such a long time for us to get to Ife, but we did, and had a conference. And they also had an institute that had herbalists, a doctor, and the pharmacists and pharmacologists all working together. So I had that exposure, which was very useful. And of course, she encouraged me to participate in local conferences. And then coaching on paper, that's a different ballgame. And she helped so much for me to learn from this. And she allowed personal development. And that is one reason why I could move from the medicinal plants as applied to humans to medicinal plants as applied to insects. That was what I did. I moved there to use insects as a model. But today, I have had to come back and use medicinal plants applied to humans. Now, kombucha. And I want to highlight all these in my presentations. So I had mentioned these are some students that she mentored. These are plants that she worked on. Eustace mentioned some. So we have Cryptolepis used for malaria, Desmodium, which we are talking about, not only used for asthma, but also menstrual pains, and also for lactation. Then Thonningia, also for asthma. Ocimum canum, akokobesa, for diabetes, Indigofera arrecta, adenia for hypertension. Question is, well, how did she go about this? So I just want to illustrate this with one specific one, and that is the anti-asthmatic plant, Desmodium ascendens. So the first question she would want asked is, what experimental model to use? And so we had to decide on that. And being the first student, you can imagine, those days, there was nothing like computer. If there was, at least, the kind of computer we had was a very huge one, which was in physics. That was what I used for my analysis anyway. But it wasn't the kind of computer you can sit here, key, and you get references. No. So I had to go to BAM library. I had to pick all these books, or abstracts and whatever. You can imagine it. And then keep going through. As long that after several months, then I came struck on this. Anaphylaxis. And Professor Okpako was making a case for use of the guinea pig ileum to be able to establish that an asthmatic condition is due to what happens when you induce that in the guinea pig ileum or lung. So because I got that idea, we discussed, and then we started. All that there is to this is that, so the guinea pig, you will inject, we use albumin, egg albumin. You inject it, and what that does is to sensitize or make the guinea pig, just like you will go and take pollen if you are sensitive. That pollen will now make you reactive the next time you encounter that. So it's a similar thing we are trying to imitate here. So we sensitize the guinea pig, induce it with egg albumin, and then we will isolate the ileum or the small intestine from it, use a bath, and connect it to the camograph or the physiograph I was talking about, and an amplifier or transducer will, whatever constrictions or contractions take place, it will be recorded. And if you do that, if indeed a plant like Desmodium ascendens works, what it means is that it can suppress that contraction. Because in asthma, it's this constriction that makes it difficult. Then you find it difficult to breathe. So once it suppresses it, then it means you can breathe. So here, we put that in, and then we apply different quantities of the plant extract and in the bath. And when we do it, then we generate. So this is what I got from the physiograph. At 9 compared to the control, which is where you don't have any extract, you can see it's a higher peak compared to when I introduce a concentration of 4.5. Then when the concentration goes to 9, you can see 9 is much smaller compared to the 4.5 and the control. So that's how we were studying all this. Now, so I'll come back to this. A lot of so we can study whatever we want, the model. We can use the whole animal and study whatever we're desiring inside the animal, or we use a test tube, we use a bath, or we use whatever, which means outside the insect. You see? Because outside the animal. So we, you do that with the isolated preparation, like the intestine in here. And now we call that in vitro study. So what I've just showed us, we got inhibition, and which suggested that the extract could prevent asthmatic attack. You all agree? You are not convinced? And so we went on, after I was gone, the next group of students who worked on Desmodium used, administered, instead of isolating the tissue, which is the intestine, and administering, they actually administered the Desmodium into, gave it to the guinea pig. And as it drank and it was sensitized, when the tissue was taken out and the same study was carried out, the result was the same. And indeed, even for lungs, it was the same. Thereafter, the next questions, biochemists, we are not satisfied with just surface, We go beyond and want to get to the root of everything. So the next thing is, if this is happening, what in the plant is causing it? And how is that happening? So the mechanism, we want to look at all that. So what in the plant is? So from 1987, that actually was what Prof Addy began to do. With all the students she had to work with, who he had to work with, they isolated fractions. They work on a number of what we call pathways as biochemists in our system. When we are talking about respiration, we can talk about our taking in air, our breathing out carbon dioxide. But that's not all. Within the cell, what is happening? What molecules are involved? We are desirous to look at all that and look at how they interact and trace that. And the cause of that, tracing that, is what we call pathway. So arachidonic acid is one of the systems or molecules that are involved in inflammatory responses. And it's made through the action of some enzymes. And we refer to these groups of enzymes as cyclooxygenases and lipooxygenases. And Auntie Marian, if you permit me to call her that way, show that fractions and isolates inhibited these enzymes. And so these are some publications. You can see effect of fraction 3 on contraction of respiratory smooth muscle. Then you have the fractionation we're talking about is separation. And we use different means. So one of those means is chromatography. There are other means by which we do our separations. And we do those separations until we come to the actual substance, single substance, or maybe a group that effect whatever response that we see. And so here, she eventually isolated triterpenoid saponins as one group of substances which had a potential anti-asthmatic effect. And so now you go to Mampong, and the place is very well established, not like those days, and they are producing Desmodium, which is, contains the Desmodium for managing asthma. Now I want to move to kombucha. Having laid the foundation, the basic, what we have done was to use animal model. I could have gone on and given other examples. But you will see the few examples I will also give using the kombucha. I want you to see how the training that Auntie Marian gave me, the interactions that we had, how it has impacted what I'm doing currently. Had it not been for that, I don't think I would have gotten into this kombucha research. At any rate, when I was in those days, second year, I didn't like microbes at all, and kombucha involved microbes. I got grade C because I didn't want to learn it. But today, I'm working with microbes. So the students here, they are hearing it. So the aim of the presentation on kombucha is to provide scientific evidence based mostly on animal studies to support some claims to its efficacy, and awaken interest in the use of kombucha as a choice drink. These days, there are all kinds of power drinks around with a lot of sugar. So if we can replace it with this, it will be good. And stimulate interest in further collaborative scientific research. Some of us are trying here, but I believe we can do with a few more collaborations. And then to engender interest in the development, production, and commercialization of kombucha. Why am I saying this? In 1987, precisely 15th of August, there was this publication, Fungus That Prolongs Life. And it was on the news. I don't know how many remember and how many saw this. And so I was then in the in UK, and my wife came and had brought a flask containing something. And she said this thing was what saved her life because she had sores all over her body, in her mouth, and she went to the dermatologist. She was going around and nothing, and God spoke to her that use this, and she did. And it cleared. So she brought it. And from then on, so I'm talking about 1988 till today, we use it in our house. So when it looks like the Ministry of Health intervened together with Pharmacy Board and said, oh, no research has been done on it, then people threw it away. And that was the end of it. So when we came, we were asking people. At a point, our culture was getting bad. And we were asking, we couldn't get any. Mr. Fei is here. And eventually, we were asked to go to food research, and that there's somebody there who is working with it. So I went there, and lo and behold, he was culturing. So I got some. He also happened to be an osopho, so I got. So I got it, and And that was the first time I had actually seen this growing in the way I saw with him, because ours was just some small thing. So we continued using it. And then in 2008, the thought just came. I felt, well, I'm retiring. And I thought nobody was doing anything after 20 years of declaring that this thing, they were going to look at it. So I decided, let me look at it. And that's how come I started. And we got, I couldn't get money. But fortunately, Dr. Oklu, now Professor Oklu, Dr. Michael Ofori, these two worked together with me, and we got a master's student and looked at biological activity generally. And lo and behold, we were seeing positive results. So we said, well, this is something we can follow up with. And so in 2011, when we were moving, thanks to Dr. Saki for pushing for us to get this place, I approached Professor Oklu, and he gave a room. And we started using that as our lab for preparation. And we, since then, have been carrying out research there. Surprisingly, whilst we were struggling with whether to accept this or not, in the US, it went there in the 90s. But they, as soon as the thing came, they analyzed, they said there were no pathogenic organisms. And then the decision was, while no direct link has been proven with regards to health, they want consumers to use it, but with caution, while they worked on it. And so at that time, there were no regulations. But now, kombucha is a big thing in the US. Multi-million-dollar business in the US. And they are regulating it because people have tried to infuse it with alcohol, higher levels of alcohol. And the US, the FDA has clamped on that. So now let's go on. Well, quickly, so what it is, you have a floating cellulose, pancake-like culture. It's cellulose, it floats, and it's on fermented tea. So you have tea, sugar tea, which you introduce these microbes onto, and they ferment it. And once they ferment it, it becomes useful beyond what tea itself is and beyond what sugar is. We can also use other plant-based materials to start, and we have done that. So, well, this is the mat, we call it, and that is the cellulose with the bacteria, safe, non-pathogenic bacteria and yeast hidden in there. And the implication of that, so here, we can classify it either as a probiotic. And those of you who know probiotic, very useful now. It's one of the things catching. And nutraceutical, we all use food supplements, most of us, I will say, or as a functional drink. We all use things like yogurt and so on. Now, the broth, of course, is what we drink. But you can also use it as a stain remover. So if you have stains on your tiles, just put a little bit and it will clear. Now, the interesting thing about the mat, first of all, look at this. It's being studied for space exploration. So the mat with the microbes for space exploration. Food packaging. I was listening to BBC. I think it was in October last year. And they have started developing kombucha as packaging material. Kombucha mat as packaging material. It's used for clothing, cosmetics. In Brazil, it's been developed for plastic surgery. And they also use it for animal feed. So here, those who know relish, they import it from Germany. And 3 years ago, we were trying to compare that with ours. They were selling it for 140 cedis. Ours, the big gallon here, is 25 cedis. And it's also been formulated as capsules. Now, there has been review of this drink by a number of people because it actually has existed since the claim is 220 BC. That's how long it's been in existence. And a lot of it, the publications, earlier publications, are in Chinese, are in Korean, Russian, and so on. And how many of us can read that? So we don't have access to it. NS in 2003 provided some clinical evidence. He was looking for clinical evidence, and he found none. And Jaya Balan, he, in his review, looked at animal experiment and cell lines, and recently, this man and his group, CAP, they searched about 310 articles in English and got only one human subject. I got one additional one. So none of these human subject literature actually have made proper claims. The kind of work they did, when you are talking about clinical trials, it's not really the kind of clinical trials that will qualify it for acceptance as a drug. And it is not a drug anyway. So the fact that a lot of space is being dedicated to it, at least, should make us be interested in it. Now, our work using molecular tools has identified all these bacteria and this yeast. And these are yeast and bacteria that basically are involved in making beer. That doesn't mean, but beer, yogurt, and all this is the same group. So kombucha, if you drink these things and you eat, you use them, it means that you can also use kombucha. So here we are. To illustrate, you have tea, you have sugar, you have your yeast and bacteria in this, and it gives you your kombucha. For those who are interested in reaction, here are the enzymes involved and the reaction. Now, so you can see here antioxidants, very important for us. And we have the chemical constituents in kombucha include sugars. We start with sucrose, but much of it is broken down to give us some glucose, some fructose, and acids. When I say acid, I mean good acids, so don't worry. So we have amino acids, the organic acids. These are the good acids, particularly this one, which is used in removing toxins from our system. And then we have polyphenols. Much of the antioxidant activity we are talking about rests in this. And then we have minerals, zinc, cobalt, and so on, which we found, vitamins, And then there are a lot of enzymes in there. So the history, I think I've talked a lot about this, but it spread so from China, Russia, and then, now those are the countries. You can see Ghana, we haven't marked it yet. Right, the claimed health benefits and what I call biocharacteristics. So a lot of claims that it stimulates the immune system. And we want to believe it's because of its activity in immunomodulation. You will understand. Then it improves resistance against cancer. And this is because of the immunomodulation and then antioxidant activity. In fact, talking about this against cancer, since we haven't tested, let me just tell a story. In Russia, the Academy of Sciences in 1951 did a research because they realized that there was a lot of problems with cancer. So a group was asked to investigate. And they went to several places. One place was an industrial area. And they realized in spite of the industry, in spite of the toxic materials, the people, when they tested them, they realized that they didn't have any cancer. So they were wondering. So eventually, one day, the leader was led by a lady to a pot which had kombucha in it, which she said they all drink. And that is what had been saving them. So it provides relief for inflammations. Auntie is here. She didn't believe in kombucha, and she had some pain on the wrists. And I asked whether she wanted to. She tried it. It worked. So it sharpens eyesight. About 3 days ago, my brother is here. He came. In fact, I was showing him the slides, and the eye had been infected, presumably. And so he was using chloramphenicol. And pain, he was actually experiencing, and we got to my office, and he was expressing it. So I said, would you want to try kombucha? He said, no problem. No, I think it was after my presentation. Then she said, oh, let me try it. So he took it home. The following morning, I asked him, how is it? He said, oh, it's okay. It slows aging. And and you can see improves the elasticity of the skin, they claim. It assists in wound healing. I've already told you about my wife's. And regulation of gastric, intestinal, and glandular activities. One of my students, our student from Liberia, 2 years ago, he came and excruciating pains, and he said he had done endoscopy and so on. So I asked, would you want to try this? He tried. Now, he is working on H. pylori gastric because of that, I believe. So I've already mentioned this, the mat. Now, so the antioxidant activity, we the men here, of course, will be concerned very much about this. The first person who found this, that the body produces free radicals. And cells, we are talking about, which does this. And these free radicals, when they are in excess, can be damaging. So they need to be mopped up somehow. And the antioxidants that we take mop these things up. Otherwise, they cause damage. So, for example, the cell, the sperm cell, they can cause damage. And this man realized that there was loss of motility and there was infertility. And all this because there was damage to the proteins within the cell. There was lipid peroxidation, destruction, basically. And the membrane also damaged, and the DNA. So all this leads to the infertility. So what we have tried doing is to establish that there was protection, at least against the lipid peroxidation, which I just talked about. We had one of our students, the first student who worked on this. Her work showed clearly. You can see we used arsenic to more or less induce the damage. And you can see that kombucha brought relief. This is the normal, which means not treated. When arsenic is used, it destroys more. And then when kombucha is used, it reduces it. Ascorbic acid is also known to be an antioxidant. And you can see that even our 2-week-old kombucha is doing better than ascorbic acid. So we compare that with tea, and you realize the kombucha is more potent. The smaller the value, the more potent. So kombucha is 3 times as potent as the tea itself. So it's not just the tea which is doing it, but the fermentation has introduced something in there. Now, except that, of course, compared to BHT, which was our standard, this one was more than 10 times higher than ours. But at least ours is an extract, and you can just keep drinking. Now, so we realize, if you look here also, our polyphenols or phenolic contents within the tea, which when fermented gives us more. You can see the unfermented has this. The fermented, we are getting more polyphenols. Basically, what it means is that the bacteria and the yeast somehow make use of the enzyme to synthesize more of these, okay, and providing better antioxidant activity. What about antimicrobial? We haven't done much on this yet, but I just want to show us this study that some other group had done and indicated that these microbes indeed affect the pathogens or other microbes. So one of them is Helicobacter pylori, which causes ulcer. Okay, so if kombucha helps with that, and like I told you, this is our student who came from Gambia. He, having been cured, and he was not the only one. There was another one who also did endoscopy, had a problem, took kombucha, and she was okay. Now, one of the questions that people ask is it probably the acid which is causing this antimicrobial? In other words, has the effect? We know acids, so they can damage anything. So at least we've shown here, if we neutralize means somehow we remove the acidity. So if you neutralize, you can see what happens. The fermented, which is kombucha, for this particular microbe is given 28. This is when we neutralize. Well, there is a slight reduction, but it's not that much. And compared to chloramphenicol, so we are doing well. Pseudomonas, another group, you can see what we have compared to chloramphenicol. Here, E. coli, it's about 2.5 or so. Right, I'm sure we all will be interested in this. And I have been, particularly because I used to have a lot of malaria. Every August and July, August, I will break down and more or less be hospitalized and so on. So our preliminary studies showed 30% inhibition at approximately 474 milligram per mil for kombucha, working with field strains of P. falciparum. You will say that is too much, but well, that's the extract. But it did something compared to artesunate. But here, so more recent work, all these are still preliminary on DD2 falciparum. The kombucha gave 5.2. The neutralized kombucha gave 3. And artesunate gave 0.1. It's not bad. For those who do not know Buruli ulcer, my colleague here works, the head of department, Dr. Mosi, works with Buruli ulcer. So we definitely can't do kombucha work without working with Buruli ulcer. So Buruli ulcer is caused by a mycobacterium. So it's also a microbe. And there is this toxin from it, which causes the kind of thing you would rather not see. And for that reason, I would say viewer discretion is advised. And so these are the stages of Buruli ulcer. You have a nodule. It's not known yet how it's transmitted. It's not clear. But a nodule comes, then it develops into a papule, then a plaque forms, and then this terrible one. All due to this. And we initially, when we started, we thought, oh, kombucha will do something to it, and then we'll have some big recognition. Unfortunately, it didn't, but at least it did something. It did something. So mice were fed for 30 days prior to inoculating or inducing it with the mycolactone, that toxin. And we found that there was delay in the manifestation of the Buruli ulcer. At least that is something. So the evidence is this. Here you are, the swelling, you remember all those, the nodules, the steps. So you have the swelling with a slight reddening. Within 18 days, it appeared with the controls. That is no kombucha. But by then, nothing has happened. So there is a delay. And the reddening and swell within 28 days. And it took 56 days before that happened when it came to the one which had been drinking or had had kombucha. Then the open wound, 35 days. But still there was a delay. It took 64 days with kombucha. And then the ulcer, 50 days in the control, which didn't have kombucha. 80 days with the one which had it. Then within 57 days, all these animals, they died. But those with kombucha were still surviving. Isn't that good evidence? That at least kombucha is doing something. And what is it doing? If nothing, if at least it's not destroying the mycolacton, at least it's offering some protection by boosting the immune system. Most likely. We have to study that. We have to look at that and confirm. Then inflammation. I had already mentioned inflammation. You can see here, again, this time it's with the same mycolacton. So it was injected in the paw of the rat. So we were studying the extent to which it will swell or not swell. And of course, because they can leak this, you can imagine this. This is the topical application. You can see from this that at least where there was no treatment, that was where it ended. Where there was treatment, oral and topical and oral only, you can see at least that the paw, the swelling diminished a lot. Then what took me to kombucha? Wound healing. That was the first of the things that I wanted to look at. We've studied this. Ours was very preliminary work, but really another group established even that kombucha's wound healing property is about the same as brucin of Bayer. You know Bayer, the pharmaceutical company, okay? But that's not what I can talk about now. What I'm talking about is what the evidence we have shown. So you have the unfermented tea. We are looking at the rate of the wound contraction. And you can see that where, again, the oral and topical, a lot more. And then the topical only also gave that, compared to the control, which is here, at least a little bit. So, of course, there are things in kombucha which would have accounted for this, including zinc, the antibacterial property, the shortening of the inflammatory phase. We've looked at something like that. And there are antioxidants, very, very important in this. This is another thing that I'm sure everyone will want to be interested in. And that is anti-diabetic property of kombucha. So, since there isn't time, you see, here it is. This is giving us the area under each of these curves, which gives us an indication of the change in the blood glucose level. So we find that when the rats were given kombucha, 5 milligram, 25, and 100, you can see, compared to, much lower, compared to these two anti-diabetic drugs, synthetic drugs I'm talking about, which they give. Metformin, glanbetamide, these are given for diabetic treatment. Look at this and look at kombucha. Isn't it doing anything? So, well, how safe is it? US, they've established that there is no problem. If you overdrink it, you'll be in trouble. Because it's not like Coke. So you have to watch how you drink it. But I'm fortunate to have a toxicologist on the team, Professor Lord Okine. And so we did these studies, the acute toxicity studies, chronic toxicity studies, and we established that kombucha didn't significantly affect body weight or the organs of the mice over the period of administration compared to the control. And for example, this is the body weight. You can see 1,000 milligrams per ml. So it helps also in controlling your weight in a way. And we looked at histopathological, we did the histopathological studies and found no significant effect on kidney morphology at this 1,000, no effect on lungs, liver, and heart. And so we can safely say that 1,000 milligrams, which is equivalent to 200 ml of kombucha. And so those who want to drink will give you a bottle, 200 ml, to take along, at least. So the average consumption is about 200 ml once or twice a day, which is about 1 gram. So we can safely say kombucha is safe if drank in moderate quantities. I want to conclude here. What so far I have presented reminisces Prof Addy's legacy of efficacy testing of herbal preparations with animal models, isolating the active components and determining the mechanism of action. And I've adduced evidence to show that the use of this approach does provide scientific basis for the numerous health claims that are attributed to kombucha. And we can also safely say that you're taking 200 ml once or twice a day could be helpful. At least it could boost your immune system. I want to make some recommendations. First, it's clear that we can't do this alone, even though WACCBIP is grown. If I say WACCBIP biochemistry, together with WACCBIP, we have grown. And we have people from all over Africa who can help. But we still need collaborators to look at microbiology and biochemistry of fermentation and production, the health benefits and production and uses of the mat. That is one thing that we've not looked at. In fact, we started 2 years ago. I started some work with engineering on the mat. Now, so there is also the need for the regulatory agencies to set standards for both local and imported kombucha. Now, when I started, I presented a picture of then. Now, I want to present this, which is the now, where Prof Awandare and where is Dr. Mosi? Where? Yeah. So we are here. And Patrick, Dr. Atta, the young ones, I'll call them, they came after we more or less had left or were leaving. Because biochemistry, we had a 20-year gap where there were no returnees, but there were people who were leaving. So what it meant is that from them to us, whilst we were retiring, they were more or less just beginning, which is not good for us as a department. But then what is good is that when they came back with some vim, some energy, some real dynamism with ideas. And within some 2 years, you see where we are. This building is part of it because they put up a proposal that got a World Bank grant. Not small, 8 million. And with that 8 million, it was meant to train not only Ghanaians, but students across the continent. And so we have people coming from all over, including faculty members who are international. They are on they join us to train these students. And I want to thank our young ones, Prof Awandare, Dr. Mosi, all of you, for all that you are doing to ensure that, for me, now I believe that kombucha will go places. The research people are doing elsewhere, we should be able to do it here. And we should be able to publish this so that we also would develop kombucha as a product, a local product that can be used in place of the kinds of things that are being bundled around. I want to end here. Thank you. Before you come, I have thanked you, and I want to thank the family for all that they have been doing for Auntie Marian. I mean, I am highly impressed. Now, thank you. And Uncle Oko, thank you. And Auntie Jane is not here. She is also our paddy. And Mommy, she frequents her place almost every now and then, weekly or every 2 weeks or so. So, thank you. A number of my brothers are here. Our former moderator, who is my brother, FH Commander Gbewonyo. And then another brother who was the director of development of this institution during Kwapon's time. He is sitting at the back. Also, my elder brother. I have my senior clerk here to grace the occasion, Reverend R.M. Ati. So thank you all, and my sister-in-law. I can go on and on.
Ewurama's younger sister is Auntie Betty.
Betty. Oh, Auntie Betty, where? Oh, so that's Auntie Ewurama's younger sister. And I think the current parish priest of Anglican, isn't it? Ah, thank you for coming. Yes. Thank you.
Thank you very much, Reverend. Please, before, yes, we would like to re-invite the high table. And whilst they're coming up, I would like to, again, warmly welcome those who joined us whilst the lecture was ongoing. Thank you so much for coming, everyone. Yes, please, Reverend has a very big thing that he omitted to do. Please, if you want to go home safely, collect.
the person who introduced me to kombucha, Mrs. Lydia Gbewonyo.
He values his life. So the next item is for the chairperson to give his closing remarks. But before that, I think I noticed Prof Alexa Dodu walking. He's also one of the students of Professor Marian Ewurama Addy. If there are any others, please, we'd like to acknowledge your presence here. Prof. Thank you very, very much. Mr. Chairperson.
Please join me in congratulating and thanking Dr. Gbewonyo for this wonderful presentation. Thank you very much. Please, Doc, make sure that I am reserving the kombucha. It's very important. I'm not joking at all. It's a very excellent research that you are doing, and very, very relevant to us as Ghanaians. And I entreat that we take such research very seriously. Biochemistry, cell molecular biology, I know you're doing very well. You have a lot of female faculty staff on your staff, female faculty. And also, I think nutrition and food science. The biological sciences in general, yes, we are doing very well. And I think as a college, we should also do well to increase our faculty, our female faculty. I know that it has a way, it reflects on even the student numbers. If you look at the, I think, family and consumer sciences have the largest number of female students, about 90% of their student population female, followed by biological sciences, which is around 50% female. But if you look at the other, you consider the other programs, all of them are less than 20%. And overall, the female student population within the college is just about 30%. And I think we should do something about it. For the next admissions, we are going to introduce some kind of affirmative action to increase the female student population. We have also the college, I'm pleased to announce that the college have received some funding to support girls in STEM. So for next academic year, we are going to give scholarship to female students who take up STEM programs. Professor Ewurama Addy, I did geology and mathematics statistics. So during my stay here, I didn't know him very well. I only know him as the quiz mistress, but I hadn't interacted with him. I think my first interaction was when I joined the faculty in 1998. At that time, I think she was coordinating the environmental science program, and she wanted somebody to teach the environmental geology course in the environmental science program. So she contacted my head of department, and my head of department nominated me. So I came to see her, I think, at the old biochemistry building in where the, what is it, pharmacy is, yes. I went to her office. She was very down to earth. She called me, sat me down, and asked me a lot of questions about why I took the faculty position, and really advised me on how to progress in the academia. I will not forget that. She really contributed to my development, my personal development in this university, and I'm very grateful to that. So on the basis of this, I want to thank all of you for coming. And Doc, as I said, I will need some of the drinks. I'm very serious. So thank you very much.
Thank you very much, Mr. Provost, for first agreeing to chair the function and for your very pleasant remarks. The next on the item or the agenda is for us to receive some words from the family of Professor Marian Ewurama Addy. So, family remarks.
Hello, and good evening, everyone. I don't know what more I could really say that hasn't already been said, dating back to the first memorial, and then also today. Just about everything that's been talked about, touched on, even looking around the room and with the stories is exactly what she embodied. I was actually looking at some of the, I know this gentleman as Uncle Seth because I don't know if you remember the guinea pig. I used to be in the lab with my mother with the guinea pig and wanting to take the guinea pig home because I didn't want her to harm the guinea pig. So I was banned from the lab for a while. But just listening, looking, hearing from most of you, it's truly a reward, and that was a pun intended, because it's in the giving is where you get the reward. So. Very simply, on the behalf of the family, we would like to extend a deep appreciation to you all. We're grateful, and that's it. God bless you.
Without much ado, can you please cue the tune and we will stand and sing the university anthem, please? It's at the back of your program.
University of Ghana, the nation's hope and glory, the place that bears the star of peace and bids its soul to do wonders in the praise tower of learning. Is my both young and old, may we proceed in unity to uphold the public cause. Arise again, defend the cause of freedom, proceed into the integrity to make a nation proud. Proceeding to the integrity to make a nation proud. We ask for strength and wisdom as we climb the hill of learning. May we excel whatever we do as we prepare to face the world with the minds ready at all times and the conscience we to feel. May we proceed in unity to oppose the public cause. Arise and proceeding to find integrity to make a nation proud. Arise and defend the cause of freedom. Proceeding to find integrity to make a nation proud. Arise and defend the cause of freedom. Proceeding to find integrity to make a nation proud. Arise and defend the cause of freedom. Proceeding to the integrity to make a nation proud.
Thank you. Thank you so much. Please make your way up to the first floor of the main biochemistry building. The cocktail was proudly sponsored by the family of Professor Marian Ewurama Addy.